you program.
Okay.
And then I'll try to cut off the questions at 11, sorry.
Sounds good.
We'll see. Okay.
Okay.
And 40, if I say 40 minutes, is that
Yeah, that's good.
okay?
Yep.
Good. Okay. Good morning, everyone. I am Professor Allison Brooks. I am the co-director of graduate studies for the PhD in Human Paleobiology, where Sims is a student. And I wanted to welcome you and thank you for coming to the defense of her proposal. This may sound strange to those of you who aren't familiar with our program, but this a proposal for funding your PhD research is an essential part of our process to becoming a candidate for the PhD, and successful completion of this stage and the comprehensive exam makes you a candidate for the PhD and eligible to receive it. Okay, so a bit about Sims. She came to us from Clemson University in 2021, where she majored in animal and veterinary sciences. And even before she was here, she was a volunteer at the zoo and has been very involved there in outreach and public engagement. And I should also introduce her committee, in addition to her advisor, Professor Carson Murray. We are joined by Francis Subio, who's a professor of speech, language, and hearing sciences, and is also a member of our program, and also by Robert Seyfarth, who is a professor of psychology at the University of Pennsylvania and also an expert in primate behavior. And Sims will talk to us for about 40 minutes, present her proposed PhD dissertation research on the impact of early exposure to violence on wild chimpanzees. And like many topics on chimpanzees, this has major importance and implications for looking at humans. So with that, just to explain how this is going to go forward, she's going to talk for about 40 minutes. And then we are welcome to ask questions if you're not on her committee. Her committee should wait because there'll be a separate session afterwards with the committee itself to perhaps ask more specific questions. So anybody in this room or on Zoom who's not on her committee is welcome to ask any questions that occur to them. And so I will cut off the questions at about 11 o'clock. So with that, I turn it over to Sims, who is going to talk to us on this topic.
Thank you, Dr. Brooks. Okay, good morning, everyone in the room and on Zoom. I think one of those cameras is on me. Thank you guys so much for joining me this morning. Like Dr. Brooks said, I'll be exploring my dissertation project on the impacts of early exposure to violence in wild chimpanzees. So let's get started. So just a little roadmap of what we're going to be talking about today for those of you who haven't attended a proposal defense before. I'll start with a little introduction on the topic of violence and adversity, move into what I hope to accomplish with this project, including 3 aims. Then I'll talk about the significance, why this project matters, and then some broader impacts, public outreach stuff that I'll be doing to disseminate the results of this project and communicate its meaning. Okay, here we go. I want to start off this presentation with a small content warning. I know that the topic is violence. You guys came in here knowing that, but I wanted to give a little heads up that I'm going to be discussing some difficult topics in the background information section. Any photos of any violent or aggressive events before will have a little red eyeball on the slide. If you feel uncomfortable or need to step out, that's totally fine. Do whatever's best for you. and I'll just be discussing these events for academic purposes. So, starting off on a light note, we're going to have a little story time. Bet you didn't think that's what this was going to be. This is a picture of my grandfather, William Kenneth Patton. He was born in 1927 in Middle Tennessee to two hardworking parents. And he passed away in 2015. He was 87 years old just before his birthday. And what was really great to me about my grandpa, other than him being my grandpa, I always thought it was so interesting how he grew up in the American Great Depression. His behavior, mannerisms, adult choices were always so calculated, calm. He worked at the same job for 35 years, ate the same plain Cheerios for breakfast every single day, plain turkey sandwiches for lunch, almost very predictable in his mannerisms. So I always kind of started to wonder if perhaps this early exposure to food or financial instability, insecurity had any impact on how he grew up and became an adult. So this project is really inspired by my grandpa, the behaviors that he had, and how things that we experience in early life can impact our adult behaviors. Small additional funny story, my first words as a child was shoes. Clearly no impact on my adult behaviors whatsoever, but that's kind of what the basis of this project is going to be about. So how did the environments we experience in early life impact our adult behaviors and health? So early life adversity, as defined by the National Institutes of Health, is environmental exposure that requires significant adaptation physiologically or psychologically, and that represents a deviation from the expected environment. So like my grandpa, growing up in the Great Depression, experiencing food insecurity as a child is a deviation from the normal environment and totally impacted his health and behavior growing up. So this is how I'll be defining adversity for this project, and kind of how we'll be exploring the different outcome areas. So what we know about the early environment is that it's plastic. So we know that a lot of development occurs in gestation, so when we're in the womb, which is great, but animals like chimpanzees, humans, and elephants, things that have long developmental periods, are actually more influenced by their postnatal environment. So there's lots of opportunities for growth and development to occur postnatally, and that can be influenced by the environments that we experience. And these environments that we experience can have impacts that last all the way into adulthood and sometimes even intergenerationally. The first area of early life adversity that we're going to be talking about is ecological. So this just means kind of like what my grandfather experienced, something that's different in your environment in terms of food, nutrition, anything like that. And we have really unique studies from both human and non-human primates to talk about this. The first one, perhaps you've heard of it, it's called the Dutch Hunger Winter. And this was a period during World War II when the German forces placed an embargo on the Netherlands. And the people of this region went from having normal daily calorie consumption, 1,500 to 2,000 calories, all the way down to 500 calories a day. And this was for a period of 6 months. So for the Dutch Hunger Winter study, individuals who were gestating, lactating, or early children during this time have actually been followed all the way into adulthood to see how they grew and developed as a response to this early life nutritional scarcity. There have been really significant physiological and psychological impacts, including obesity, cardiac disease, diabetes, shortened lifespans, and even evidence of increased chances of schizophrenia. What's really unique about this study is because it was a long time ago, we've actually been able to follow the offspring of those exposed. So we've been able to see those intergenerational effects. And even then, the offspring of those who experienced adversity exhibiting some of the physiological symptoms. So this is a really unique opportunity that in history that's kind of shown what the effects of ecological adversity are. So like I said, in non-human primates, we have examples of this too. We know that wild primates experience periods of drought. Even at our own field site, Gombe, there's been some periods of increased rain and then decreased rain. And some of the same effects can be seen for our non-human primate friends. So exposure to drought in early life specifically is associated with decreased survival, so a shortened lifespan, in several different non-human primate species. And even in instances like the Cayo Santiago macaque population who experienced a massive hurricane event, they are exhibiting decreased survival as well. So there's different examples of ecological adversity in non-human primates too. Shifting gears a little bit, there's kind of the second bucket of adversity, which is what I'll be putting my focus on for the rest of this project, and that's social early life adversity. So it doesn't really have anything to do with nutrition or calorie intake. It's more about what you're experiencing in your family or community environments. And there's examples from non-human primates that instances of social early life adversity are associated with negative adult outcomes. One of the most famous studies from non-human primates are called the Harlow studies in the 1960s. Just prefacing this by we don't do this anymore. But at the time, they separated rhesus macaque infants from their mothers and placed them in social isolation nursery settings for periods of 3 or 6 months, reintroduced them to social settings following those periods to see how they kind of interpreted and interacted with social mates following isolation. I'll tell you right off, it didn't go very well, particularly those who spent longer in periods of isolation early in life. They had worse outcomes when reintroduced to the social group. So that's a captive study of non-human primates, but in the wild, we have some instances of this as well, particularly in cases of maternal loss. So wild chimpanzees, wild baboons, rhesus macaques who experience maternal loss, particularly before they're weaned, which is really important for their nutrition and social integration, they have decreased survivorship if they're orphaned. The trick to studying early life adversity in wild non-human primates is having a long-term field site is really important just to be able to see those outcomes in the long term. So in humans, we have a unique kind of social adversity, which is violence. This is an area of social early life adversity that is currently limited to human study. And violence is defined as behavior involving physical force intended to hurt, damage, or kill someone or something. This is our definition for humans. And this is the definition that I'll be applying to chimpanzees for the purpose of this project. So in humans, early exposure to violence, which for the rest of these slides will be abbreviated as EEV, has one of the strongest associations between early exposure and negative adult outcomes. More than any kind of financial insecurity, more than parental divorce or disruption of the family unit, exposure to violence has the strongest associations with negative adult outcomes. So this is something that's currently unique to humans, but if we know that it has such meaningful impacts in humans, why aren't we exploring this in non-human primates? That's where I come in. We have some examples, a little bit more of this from historical situations, kind of like when we were talking about the Dutch hunger winter. There's examples of exposure to violence in research on historical events, beginning with actually the US Civil War. Offspring of Civil War soldiers who were in war had decreased survivorship compared to offspring of soldiers who were not prisoners of war, particularly if the offspring was a son, which is interesting. And in World War II, during the Blitz period, where German forces were conducting air raids over Great Britain, children of both sexes exposed to these air raids experienced symptoms of PTSD and anxiety all the way into adulthood. So we have all this evidence from humans as to why violent exposure is bad for adult outcomes. And this project seeks to explain or understand if the same outcomes are associated in wild chimpanzees. So this is kind of the big research project going forward. How does early exposure to violence impact chimpanzee outcomes? Kind of like I've been alluding to in the introduction, I think that chimpanzees are a great model to answer this question. Because we know that they actually exhibit increased levels of violence compared to humans and other great apes. Males in particular participate in what's called border patrols of their community, where on the off chance they engage someone from another community, it typically turns into lethal aggression. We know that infanticide occurs in chimpanzee communities. So we know that violence is happening, but we're not exploring what the outcomes are of that. Something else that's really interesting about using chimpanzees as a model is that in humans, when you're exposed to any kind of adversity, there's often these things called buffering factors, which are things like access to medical care, access to community support networks, even socioeconomic status can kind of mitigate some of the effects that you would experience from adversity. But conducting this kind of research in a chimpanzee model where there is not really any socioeconomic status, we have dominance rank, but not quite the same. We don't have medical care for them. These chimpanzees perhaps provide a more clear picture of how violent exposure could impact adult outcomes. So at Gombe, which is the field site I will be conducting this research at, we are lucky enough to have extremely long-term behavioral health and demographic records. Data collection began in the 1960s and is currently occurring now. And we have two unique periods of long-term violence at Gombe. I'll go into them a little bit more in detail. But these are not just short instances of violence. These are instances of violence that were continual over periods of 4 and 14 years. So a little bit about the study site. Perhaps you've heard of it. It's called Gombe National Park. It's the longest running chimpanzee field site in the world. It's located on the western border of Tanzania. It backs up to a lake. And we currently have 3 communities at Gombe. 2 are habituated. You can see on this map here, Matumba and Kasekela are habituated. Kalande is not. But many, many years ago in the 1970s, there were actually 4 communities. 4 to 3. What happened? You may be asking. Well, let's talk about it. So, behavioral and demographic data collection for this project, reliably, we're going to be using starting in 1974 all the way up and even until now. We have several different behavioral data sets on infants, mothers, and adult males and females. And we have health data, including fecal and urine samples from the Greater Gombe Ecosystem Health Project that I can use for historical health data collection. And I will also be collecting novel data collection beginning in the fall of 2024. So our 2 periods of prolonged violence. There are 2 different kinds of violence that I'm going to be talking about for this proposal. The first one is called inter-community violence, like you have an intermission in between a show. It's between 2 different things, so 2 different communities, inter-community violence. And the second one is called intra-community violence, and that just means violence within one community. So for this example specifically, the individuals of Matumba are committing lethal aggression against each other, not to a neighboring community like in the chimpanzee war period. To explain them a little more in detail so you believe that this actually occurred, it sounds hard to believe, but it's actually colloquially referred to as the chimpanzee war. And beginning in 1974, we can see this Kasekela community right here in the middle. And to the south, the neighboring Kahama community, which was much smaller. But beginning in 1974, the Kasekela chimpanzees began moving into the Kahama community, and they actually killed 5 adult males and 1 adult female over the course of this 4-year period. The adult female specifically was actually attacked 4 times, and on the last attack, she was killed, and her 2 daughters witnessed these lethal aggression attacks and were then absorbed into the Kasekela community. So we know that this period of violence occurred and that there were significant deaths at Gombe, and now that community doesn't even exist anymore. So we're going to be using the individuals who were juveniles or infants during this war period and following them throughout their lifespans, both behaviorally, physiologically, and developmentally, to see how this war exposure impacted them. Now, the second period is a little bit more recent, and it's occurring at the most northern community, the Matumba community. This community is habituated, but reliable data collection didn't actually begin until 1994. But beginning in 2004, we see what I said earlier, intra-community violence. There's one adult male in particular up here in this top picture. His name is Edgar, and he has killed 6 other adult males within his community, including his brother. So we see right here on this timeline, in the winter of 2004, we have what's called an alpha turnover event. This just means that the male chimpanzee who was the top of the dominance hierarchy was overthrown. And right here in 2004, that was Edgar's brother. He was in charge of the Matuma community, but in 2007, Edgar killed him, and now he is in charge of Matuma community. And what's really unique and timely about this project is just as of last week, our amazing field assistants at Gombe sent us an email saying that the adult males remaining in Matumba after this period of violence are starting to move into the Kasakela community. So violence may be on the horizon again, and we will have a front row seat just as these two communities become closer and closer. Perhaps another war is forthcoming. So this project is going to be broken down into 3 what's called aims. And these are the 3 outcome areas that I will be exploring as response to this violent exposure. So we have, oh, sorry, the little screens above it, but that word, it says development under there. Our second aim is health and survival, and the last one is behavior, and I'll explain them more in detail. To talk about our sample size, like I said, we're going to be focusing on the chimpanzees who were infants and juveniles during these 2 war periods. For the period from the 1970s, we have 32 individuals, and I've broken them down males and females. And then we've also broken them down by age category. So were you experiencing these violent events as an infant? So a chimpanzee who's not yet weaned, less than 4.2 years old. Or were you experiencing them as a juvenile between 4.2 and 10 years old? Or unfortunately, were you experiencing this violence in both developmental periods? So we think that period in which you experience the violence could have an impact on its outcomes. So we'll be exploring that further in the results. This is the kind of data that we have for each of these violent events. Like I said, there's those 3 aims: development, health and survival, and behavior. Because Gombe is such an amazing long-running field site, we have behavioral and developmental data for all of the individuals in these exposure categories. And we also have survival data, so just how long are they living, pretty basic lifespan. And for the Matumba community specifically, because these individuals are, a lot of them are still alive, we can collect novel physiological data to see how health outcomes are affected. So we have already collected urine and fecal samples, but we have the opportunity to collect more, not only to see how they're developing as adults, but how this perhaps violence on the horizon is going to be impacting their health as well. So our first aim, development, basically just how are the chimpanzees growing up? Are they reaching developmental milestones? Are they having shorter lifespans? How are they growing and developing as response to this violent exposure? And we're going to accomplish this aim in a relatively simple way, which is creating what's called a developmental timeline. So to frame our predictions and outline these timelines that we're going to be creating, we're going to be working with 2 life history frameworks. And a framework is just something that makes predictions based on what you experience in early life about how you're going to develop and survive into adulthood. So I'll just explain the frameworks briefly, and then you'll see the predictions and be like, yes, that makes sense. So the first one is called the developmental constraints hypothesis, and this proposes that when you experience adversity early in life, this is a really stressful condition and stress physiology that's associated with that. Decreased immune function, increased inflammation, release of stress hormones. This all will result in decreased survivorship. So basically, shorter lifespan compared to individuals who are not experiencing this violent exposure. Now, the second framework, a little bit different, perhaps a little more complicated. It's called the predictive adaptive response hypothesis, sometimes referred to as the silver spoon hypothesis. So this alternatively predicts that if you experience adversity and all the associated stress reactions, if adversity occurs again in the future, you're actually hardier to it and would have better survival compared to your peers who did not experience adversity. So if I am a baboon and I experience drought early in life and drought occurs when I'm an adult again, I'll be hardier than my cohort members who didn't experience drought. They won't really know what to do in a drought condition. However, if in the predictive adaptive response model, if you don't experience adversity again, it just basically reverts back to developmental constraints. So you're not really being hardier to anything as an adult, and you'll have decreased survivorship. So these two frameworks predict two different speeds of development. Developmental constraints predicts a decelerated developmental timeline. So because you're experiencing stress, your body's releasing all those stress hormones, you're not having time to grow and develop, and you'll actually reach puberty, first offspring, all those milestones later in life. Alternatively, predictive adaptive response hypothesis predicts accelerated developmental timelines, because if you're anticipating that you're going to have a shorter lifespan as a result of adversity, it's advantageous for you to start having offspring earlier and earlier to increase your fitness. So these two frameworks have two different outcomes and two different predictions for development speed. These two frameworks are what we're going to be using to frame our predictions for this project. Like I said, developmental constraints, decelerated developmental trajectories. And this is actually what we see in some studies of non-human primates. So for example, in wild chimpanzees, females who were orphaned exhibited decelerated developmental timelines compared to chimpanzee females who are not orphaned. And in rhesus macaques, this same population in Cayo Santiago who experienced the hurricane event, this significant period of adversity, they also exhibited decelerated developmental timelines compared to those who did not experience a hurricane. Alternatively, human research actually falls more in line with predictive adaptive response. So particularly in female children who experience situations of abuse or familial violence, interpersonal violence within the home, they exhibit accelerated developmental timelines, particularly in the age that puberty is reached. So these two study systems, non-human primates and humans, fall within these two frameworks and have basically competing outcomes. So this has framed our predictions for this project, and we've basically just come up with two. It will be interesting to see whether the chimpanzees in our study fall more in line with what's evidenced in human research or what we see more in non-human primate research. So are they going to have speed up development, or is their development going to be a bit slower as a result of violent exposure? Like I said, we'll be creating what's called a developmental timeline for these individuals, basically just mapping when they're experiencing these significant ages of interest compared to the average chimpanzee ages. So we know that chimpanzee weaning typically occurs around 4.2 years of age, maternal independence. So when you're spending less time with your mom, more time wandering by yourself is typically around age 10. Sexual maturity is typically reached around age 11. And then for females, first offspring is at 14.9 years old. So this is our average female developmental timeline. What do we think will happen to the females and males? They won't be exhibiting age of first offspring. That's okay. We can still create timelines for them. How will our violent exposed chimpanzees develop in comparison? So this is just a sample timeline that I made. This timeline exhibiting decelerated development would fall in line with our developmental constraints hypothesis, which is what we currently see in non-human primates. First aim, done. Second aim, health and survival. This one involves both previously collected and novel physiological data. So we've talked about how stress is bad for development. We can also talk about how stress is bad for health. And we're going to be accomplishing this aim using 3 different measures. The first one being a basic survival analysis. How long are you living? The second one being biomarkers of stress, so fecal glucocorticoid levels, and the last one being inflammation, measuring fecal levels of immunoglobulin A, which also has implications for immune function. So early life adversity is associated with decreased survivorship in a wide variety of mammalian taxa. like we were talking about earlier with the Dutch hunger winter, experiences of drought, experience of violence within the home. Those instances are all associated with decreased survivorship, so shortened lifespan compared to your peers who did not have these experiences. Just a few examples: red squirrels, rhesus macaques, and roe deer who experience early life adversity all exhibit decreased survivorship. I kind of alluded to this earlier, but stress, not so great for the body. And this is a result of stimulation of the hypothalamic pituitary adrenal axis. Quite a mouthful. We'll just be saying HPA axis moving forward. But when you're stressed, your HPA axis releases what's called glucocorticoids, cortisol, commonly referred to as the stress hormone. And in the beginning, that's okay. Let's say you are on a camping trip and you see a bear. You need that initial stress response to start running away from the bear. But if you see a bear for hours and hours and hours, or days and days and days, prolonged periods of stress are actually bad for your HPA axis and will lead to decreased reactivity over time. So acute stress is okay, gets you out of a bad situation, but stress over a long period of time, not good. Eventually your HPA axis will downregulate over time. For inflammation and immune function, we see a similar pattern, like with fecal glucocorticoids. Immunoglobulin A is released as a result of stress through the HPA axis, and it's responsible for passive immunity and regulating inflammatory cytokines. So in most mammalian taxa, increased levels of IgA are associated with decreased immune function and increased levels of inflammation. But also similarly to our glucocorticoid levels, a little bit of acute stress is okay, keeps you alive and surviving, but over a long period of time, it's detrimental to health, which is what our chimpanzees with these prolonged periods of violence are experiencing. So these predictions that we have for this project decrease survivorship overall, like we were saying with earlier in some of our historical studies and our studies of non-human primates, early life exposure, shortened lifespan. We predicted dysregulated levels of glucocorticoids. Perhaps initially as a response to the violence, we'll see elevated levels, but over time, because the violence is lasting years and years, we'll actually decrease levels as a result of that hyporeactivity. For immune function, measures of IgA within the fecal samples, we predict elevated levels of inflammation over time, just as a result of chronic stress, which would indicate decreased immune function. So how are we going to measure all this? I know this sounds like a lot of science, but that's okay. I'm a scientist. Our first simple survivorship curve, it just plots how long are the chimpanzees surviving compared to the chimpanzees who don't have this violent exposure. It's a basic survival curve. It's just telling us what are your odds of making it to, say, age 5, age 10, age 20, and so on. We'll be using Cox proportional hazard models to check for significance, and we'll also be running these models separately for sex, as we know that early exposure to adversity can impact male and female offspring in different ways. Our second methods, fecal glucocorticoids. Data collection of fecal samples began in 2006, but really ramped up in 2011. So what's really unique about this project is that we already have samples collected that were collected during the periods of violence at Matumba. And we can go and collect novel samples now that our individuals are adults and that violence is probably about to occur again. To kind of validate the results of this method, we're going to be using what's called a matched comparison. So like I said, the violence is happening within the Matumba community, but what about our neighbors, Kaskela down to the south? They're not experiencing periods of violence yet. But for measuring these fecal glucocorticoid levels, we'll be collecting and analyzing fecal samples from both communities and comparing them. So if we have a cohort of 30 individuals with violent exposure in Matumba, we'll be using them to compare with 30 individuals born at the same time in Kaskela to compare their fecal glucocorticoid numbers. And we think this will eliminate a lot of potential ecological independent variables that could muddy our results, perhaps. Glucocorticoids will be measured using basic extraction and enzyme immunoassay techniques, and will be conducted at the Smithsonian's National Zoo and Conservation Biology Institute's Reproductive Endocrinology Lab. Our last aim, similar to our last method, I should say, for this aim, similar to the second part of aim 2, because we already have fecal samples and we're going to be collecting some more, we can also measure fecal levels of immunoglobulin A. We'll be using the same matched comparison analysis between the two communities, and we'll be using the same extraction and immunoassay techniques. So this will just give us some more information about inflammation and immune function between the two groups of chimpanzees. Aim 2, 2 out of 3. We're cooking through this. It's going to be great. Our last aim is behavior, and we wanted to explore this aim just because we know that early life adversity doesn't just impact physiological health. Like we were talking about with the Dutch hunger winter and the instances of the Blitz, those German air raids over Europe, PTSD, anxiety, schizophrenia, there are behavioral and psychological outcomes associated with early life adversity. So we'll be exploring how behavior is impacted, doing basic social network analyses. So how are the chimpanzees integrating and maintaining relationships within their community? Aggression, are they exhibiting increased levels of aggression, perhaps because they've been witnessing these heightened levels of lethal aggression? And lastly, dominance rank, which isn't exactly behavior, but it has so much to do with chimpanzee health and survival that we'll be incorporating it into this aim. So in humans, exposure to violence in early life is associated with increased levels of aggression, particularly when violence is experienced within the home or within the community. Those exposed often become perpetrators of violence as adults, particularly in interpersonal relationships. This has framed our predictions for our chimpanzee study period exposed to violence. And we expect that our chimpanzees will also exhibit increased levels of aggression as a result of this exposure. For captive studies of non-human primates, like I said, we've seen some changes in behavior and aggression, but I wanted to highlight some more research from those Harlow studies of the 1960s, that the individuals who were separated from their mothers and placed in periods of social isolation, when reintroduced back into a social group, they also exhibited increased levels of aggression and sometimes inappropriate aggression. So if an individual within their social group attempted to groom them, they would respond in an aggressive way. So just some examples of non-human primate aggression also increasing as a result of early life adversity. Like I said, our predictions, we're predicting lower social integration just because if you're a chimp and you're acting aggressive towards everyone in your group, you would perhaps be less socially integrated, which could also impact your dominance rank, which impacts your health and fitness overall. So we're predicting lower social integration, increased aggression, and lower dominance rank as a result of violent exposure. To create a social network analysis, this is kind of like a map of all the individuals in the community, how they're interacting with each other in two main ways. The first one being a grooming indices. So in non-human primates and chimpanzees specifically, grooming is really important for making and maintaining social bonds within your community. It's really nice to groom your friends because one, that physical contact, release of oxytocin, and two, if there are bugs on them, you would want to remove that. So grooming your friends is a really nice way to make friends in chimpanzee communities. The second one, dyadic association indices. How much time generally are you spending with other people in the group? Chimpanzees have fission, fusion, social structures, so they often spend time in big groups, but then sometimes they prefer to spend time alone with just their offspring. So creation of these social networks will help us understand how their social behaviors are impacted as a result of violent exposure. We'll be creating these networks because they can change over time when a chimpanzee is at their prime age, which is between 15 and 20 years old. So what we'll do for our chimpanzees who survive to this age is we'll create their social networks, see who their connections are with, how strong are their connections with their group members, and how integrated into the community are they. And there's 4 ways that we can do that. The first 2 are pretty simple. How much time are you grooming others? And how much time are others grooming you? Those are in degree and out degree. And then between this is called it's called social bridging. And I like to think of it as if you're at a conference and you're perhaps an early career scientist, but your advisor is with you and can introduce you to lots of other people at the conference, they are your social bridge. So it's really great to be an advisor and introduce your early scientists to all your cool friends. And that just makes you more integrated into the community because you know so many other people. And then our last one, our measure of centrality. We know that it's good to have lots of social partners, but it's also good to have social partners who also have lots of social partners. So if I'm a chimpanzee and I spend all my time grooming someone who's on the periphery of the community and doesn't have a lot of social partners, that's less advantageous than if I spend time grooming someone who's the alpha male or the most high-ranking female who's really, really integrated into the community. So these are just the 4 measures that are used to create these social networks for our chimpanzees who will be exposed to violence, who are exposed to violence. For our aggression method, pretty simple. We're going to be creating an aggression rate per individual. So this actually hasn't been done at Gombe yet. We'll have to create an average aggression rate first and then compare with our violent exposed chimpanzees. Like our prediction said, we're predicting increased levels of aggression as a result of violent exposure. And we'll be calculating this just based on how many times chimpanzees instigate an aggressive encounter and also how many times they're the victim of an aggressive encounter. So 2 measures for this to create an aggression rate. And then lastly, dominance rank, like I said, is very impactful for chimpanzee health and fitness. And in non-human primates, low social rank is often considered a stressful condition. There's actually studies of early life adversity in captive primates that use low social rank as a condition, as an adverse condition, and there's negative associated outcomes with that. So rankings are established based on the direction of pant grunts, which is a vocal indicator of subordinates. And we'll be creating and comparing these dominance ranks based on, for males, like I said, their prime age. So Edgar, for example, my violent Matumba male, at his prime age, he's the #1 ranking male. But for females, we'll actually be using their entry ranking. So what's the rank that they enter the dominance hierarchy in? Because dominance rank is maternally inherited in chimpanzees. So I know that this project sounds like a lot, and it is, but I'm confident in my abilities to accomplish it. This is just a research timeline of when I plan to have all of these aims and methods accomplished. I'll start by actually completing a lab rotation in the reproductive endocrinology lab, just so I'll be able to run the novelly collected samples myself here in DC. And then hopefully in the fall, I will be able to go to the field, start collecting novel samples, and hire a research assistant to help me accomplish that. Lots of chimpanzees, lots of poop to collect. More hands make light work, isn't that what they say? And then starting in 2025, I will be back here analyzing, writing up the results. So check back in with me in 2026 to see the results of this project. And again, just circling back to our simple question, I hope these 3 aims have convinced you that this is a meaningful project. But how does early exposure to violence impact chimpanzee outcomes? The 3 outcome areas that we're going to be exploring and all of the methods that we'll be using. Hopefully we'll provide a comprehensive and complete look at how this violent exposure can impact health and survival. So why does this project matter? Chimpanzees do not really live near us. We don't really interact with them on a regular basis, but they are some of our closest living relatives. And this is a facet of social early life adversity that has not been studied yet in wild chimpanzees. So that is what I hope to explore. Like I said, we know chimpanzees exhibit violent behaviors, and we know that in humans and non-human primates, social early life adversity is bad, so why not explore this further? And the chimpanzee study system will eliminate a lot of those human buffering factors like we talked about earlier, access to medical care, socioeconomic status, things like that. And then alternatively, based on the results of this project, we could find that chimpanzees are perhaps primed for this violent exposure. If they're exhibiting violence in greater rates than humans, are they more used to and perhaps prepared to experience violence as adults? Are they not going to exhibit these negative outcomes that we see in humans just because it happens more frequently? So we think that this project is really unique and will provide some really interesting results into how violence is interpreted in non-human primates. So the broader impacts, perhaps my favorite section because I love public outreach. This project has significant implications for animal welfare. I think currently in the United States, there's 10,000 chimpanzees living in human care. And understanding how violent exposure impacts health and survival could inform management decisions of these captive populations. I plan to present the results of this project at various primatology conferences for both field and captive primate researchers. And then I also have been, like Dr. Brooks said, a volunteer at the National Zoo for almost 4 years now. In the ape house, I absolutely love it there, getting to interact with the public, not only educate them about non-human primates, but how they can contribute to conservation efforts in the wild. And then I'll also, in addition to continuing my volunteer service, I participate in what's called the Letters to a Pre-Scientist program, where I have a middle school pen pal, actually in South Carolina, which is where I'm from. Very cool chance that I got her. And we just exchanged letters on my path to career in science and maybe things that she would be interested in exploring further. So I love getting to do that. This project also has so many opportunities for collaboration. Gombe is funded by the Jane Goodall Institute, and we hope that by receiving additional grant funding in the spring, we'll be able to hire a Tanzanian field research assistant. We'll prioritize hiring a female research assistant, someone who's interested in pursuing a career in science, and they will have the opportunity to have authorship on all papers resulting from this project. And then within our GW CASHP community, we have some wonderful undergrads in our lab. And perhaps we'll recruit some more to help us analyze and interpret this data moving forward. Woo! So this is the fun part of the presentation. I just wanted to extend a huge thank you to everyone in this room who's come and listened to me talk for 43 minutes, and everyone on Zoom. I sent this Zoom link out to so many people, and I'm so honored that so many of you have come to watch. There's so many collaborators that we have at Gombe, including Drs. Ann Pusey, Ian Gilby, Elizabeth Lonsdorf, and Michael Wilson. I also planned to collaborate with Dr. Janine Brown at the National Zoo for those glucocorticoid and immunoglobulin analyses. I have so many wonderful mentors over the years. Some of them are on Zoom now. Woo, this is hard. And there's so many great communities that have supported me in getting this far, including my National Zoo family. I call that my mental health day of the week, just because I love spending time at the zoo with not only the animals there, but the people who work there as well. My Emory family, who's on Zoom, my first ever time working with early life adversity and non-human primates was at Emory. And then my CASHP Primate Behavior Lab, all in the room right now, and on Zoom, Tor and Sylvain have seen this project in so many different iterations. My practice talk friends who came and watched this talk last week helped me format everything, get those capital letters in order. And just the CASHP community in general, everyone in this room has been so supportive of me and my project this far and will continue to be. In my master's cohort, we all finished in May and we're such an awesome bonded group. Some of them reached out to me on Twitter and Instagram about this. And then my family, my mom, she's on Zoom this morning. Hi, Mom, who encouraged me to pursue a career in primatology when she didn't even know what it was. So love my mom so much. Logan, my partner's in the audience. He's a veterinarian, so we're all animal people here. My aunt and uncle, my sister and brother-in-law, my partner Logan's family, they're also on Zoom this morning. And then my Clemson family, Clemson's my home. I'm so loyal to all the friends that I made there and all the friends and family back in Columbia that are also watching this presentation. So special thank you to Carson, my advisor, who's been talking about this project with me since 2019. So we've come a long way. Yay! Hi, Zoom. Thanks for coming. And yeah, I'll open the floor up for questions. Ooh, take a water break first.
Will you manage the questions on Zoom?
Abby will flag them.
Okay.
Yep. Courtney. Hi, friend.
Phenomenal. I would expect to glad to you sharing. I'm fascinated. I have a couple of questions.
Yeah.
One, can I ask you?
Yeah, sure.
Can you speak up, Clinton? We can't hear you.
Sorry, yeah. Do you anticipate kind of essentially a bonding effect for some folks who were exposed together? Which may also kind of play into this precipitating of another war that is internal.
Yeah. So it's really interesting that basically we're thinking of it as the Matumba males that have survived Edgar's reign of terror, if you will, are extremely strong and clearly looking to gain territory against the Kasakela males who have basically no experience with lethal aggression. So this upcoming interaction will be really telling for how this violent exposure has impacted their behavior. Also, what you were saying with I don't want to call it trauma bonding, but like bonding through this experience. It's interesting, the Cayo Santiago macaque population who experienced the hurricane became more socially bonded following the hurricane. So they increased their social effort with their group members, which is a little bit alternative to what we would think. If they're exhibiting greater levels of aggression, why would you be friends with someone who's so aggressive? But perhaps what you were saying as a stress response could be advantageous to increase your social effort. Yeah, it's a very timely, I did not ask them to start moving into Cascala, but they did. Did you have a second question?
Yeah, just a lot of I think it's hard to parse apart major events that are collective versus individual experiences. And some of the kind of examples that we have from humans, we can see that separated a little bit more. Is that something we have for in?
Yes, so initially this project would also incorporate instances of infanticide, but it's so hard to parse apart who's actually witnessing and like an instantaneous event like that versus our two violent events that are so prolonged. We're expecting that everyone in the community will have witnessed at least one of the 8 attacks, compared to something that's like happens one time within one community. Yeah, Andrew.
Great presentation. I had a question about, it seems like, if I understood your methods correctly, that you're going to sort of define exposure to the violence as were they a juvenile or an infant during the violent period.
Yes.
But is there a possibility that there's like social buffering on how that's experienced? Like, for instance, if Edgar is the murderous chimpanzee,
He is.
If my mom during infancy was a grooming partner of Edgar's versus if my mom doesn't get along with Edgar, could that just change how I experience his murderous rage? Or during the wartime, if you were on the winning side versus the absorbed side, could that change how you observe it? And is there a way to sort of deal with
it?
that?
It could. For the war period specifically, we're including the individuals born in Kahama and Cascala. Kahama's really small. There's not many. But yeah, we'll expect that to, like you said, violence was not being aggressed towards your community, it was your neighbors, so that could impact how you're interpreting it. And we also expect, like what you were saying with the grooming partners, dominance rank could play into maybe something as a social buffer, just because typically, like within Edgar's case, the high-ranking females aren't the ones who are being attacked, but lower social rank is considered a stressful position. Perhaps you're grooming some of the males who were killed off first because you're of low social rank. So we'll have to be controlling for that in our analyses for sure. Rank is a huge predictor of a lot of things in chimpanzee research. Something to be considered. Yeah, Abby. Tor has a question. Hi, Tor. Good morning.
Hello. Yeah, I guess is it okay if I ask the question through here?
I think your findings from Gombe will compare or have the potential to be comparable to other chimpanzee populations, particularly given that Gombe, compared to other sites, is reduced female immigration to other communities, which has the potential to lead to mother-daughter coalitions. Yeah, so I don't know if anyone's watched Chimp Empire on Netflix, but it's a documentary about another field site in Gogo in which the males there have been conducting regular territory patrols in violent manners similar to ours. I think it'll be particularly interesting at Gombe just based on the long-term data that we have and that these violent events the prolonged periods are so far apart and relatively isolated. Like in that large timeline that I made outside of these two violent windows, we only have like 3 or 4 instances of lethal aggression against adult males, including territory patrols. So really not that many over the course of Gombe. So these periods are particularly unique. I can't speak too much on violence levels at other field sites, but I know Ngogo has a lot more males than we do. So that could be impacting the violence that they experience. Yeah, good question. For second question, food availability can fluctuate and also lead to adverse conditions. Does this affect the years that you're looking at? If so, how are you considering this? So, great segue into my master's thesis in which I looked at food availability at Gombe from 1974 to 2020. It's not fluctuating that much. There's a couple years of drought at Gombe, but my master's thesis explored survival outcomes as a result of that, and there were no impacts. So yes, they experienced drought, but it's typically 1 year at a time, and it's a long time before the next drought occurs again. Yeah, thank you, Tor, for coming. Yeah, Allison.
Yeah, a quick question. You alluded to differences in how males versus females react to or experience and have outcomes from being involved in violence at an early age. But you didn't specify what you might be looking for in chimpanzees. I wonder if you could expand a little bit on that.
Yeah, I think the Matumba example is particularly interesting because all of the violence and deaths have been adult males. Edgar is not attacking the adult females in the group, whereas in the chimpanzee war, adult females and adult males were killed. So I think just based on who's the victim of the attacks could impact maybe how they're experiencing it. If you're a female at Matumba and you know that Edgar's not interested in killing me, perhaps you and your offspring are not going to be as impacted by the violence. But perhaps if you're a male offspring and you see that all the adult males in your community are being offed, your reactions could be greater than if you're a female offspring. We'll definitely be running all our analyses separately by sex.
Do you have expectations, though, as you do for other parts of your study, based on the human evidence?
The physiology and survival is pretty much the same for males and female outcomes. The development, particularly for females in humans, is accelerated more so than males. The only kind of thing that I found from human research and development is that it was slower for males, but accelerated for females. And then for behavior, the aggression was more pronounced in males exposed to violence. But yeah, it'll be interesting to see. Thank you for your question. Yeah, Jack.
Do you have any predictions about like severity of things? Is it better than it lasts a long time?
Can you repeat the question? Because I think the Zoom group.
Yeah, Jack asked if I expect the 2 different violent events to have different outcomes just based on the prolonged, like 4 years versus 14 years. I think yes, not so much in the time, but if you're a chimpanzee, you know that territory patrols happen and they often turn lethal, but are you expecting lethal aggression to occur within your own community? Like that could arguably be more shocking and scary to you than if like, oh, I know that the males in my community are protecting us and we don't care about that other community versus, wow, my own family members are at risk. So there could be different outcomes based on that alone. Excellent question.
Well, thank you very much for this.
Thank you, Zoom.
Your committee will
Yes.
reconvene.
Yes, Robert and Francis, we will reconvene in this Zoom room in about 10 to 15 minutes.
Okay.
Guys,
Good.
so much for coming. Bye.